So my talk is really about the incorporation of genomic information into BREEDPLAN. It's a big area, and I'm really going to step you through a whole range of things. I'm going to try to do that very quickly, because there are a lot of things happening in this area, but also a lot of new understanding that's coming along probably in the last 10 or 15 years. And all of a sudden we do realize, in a lot of cases, we don't know as much as we hoped we know. And I think that's one of the big things that I will continue to mention, is that that's our current understanding. And there's quite a few areas that we need to do a lot more work to understand exactly what's going on. So, yeah, I don't have an introduction of exactly what I'm going to go through, but it's really just taking you on a journey from where we started until where we now, and of course then looking into the future a little bit as well, of some of the work that we're looking at doing in the next few months or year at least. So I'm sure that you're all aware of that situation where, of course, for that calf to grow into that cow, there's two major things that influence that calf. Of course, the one, and probably the biggest one, is the environment. The environment has a huge impact on how that calf will develop, but also the bit that we're, of course, interested in genetics. Because that's the part that we really can focus on and where we can make a big difference. And that, of course, whatever we do in this area, it's carried over from generation to generation. Whatever you do in the environment here influence that animal at that stage, and that's the benefit you get. And Peter very clearly also shown that with this carrying on effect that we get, we certainly can make a big impact on our situation in the long term and financial benefits there. So that's where we're starting off. So, of course, if you then look at that genetic component, you also, of course, have how do we really determine genetic merit? How do you determine which animal is genetically better than another? And, of course, over years things have changed, where people right in the beginning, I'm sure, have looked at an animal and said, yep, no, that one looks about right. And, of course, that's still the case now. We do have to look at animals even now and make sure that they are acceptable, because we don't have all of the information at the moment as EBVs. Pedigree, of course, is important as well, and for many decades, people have only really had those two, where they had the visual, and then, of course, where they keep track of pedigrees, you just make sure which lines are there. Then, in my lifetime, certainly, performance has started to come into play. So there's, of course, now a lot more performance recording being done, so that's being used as well. And then now we're starting to see EBVs, or not now, we've seen them for a while as well, but really EBVs are incorporating the pedigree information, performance information, and then give us an estimate of those animals' values and genetic merit there. We also have selection indexes, where we heard from Andrew this morning. It's a lot of the work that we're doing in that area. And the bit that I'm going to focus on really is on genetic markers. So how now that we certainly have had that for a long time, we're now into this area with genetic markers. What's the impact of that, and how accurate is that to determine the genetic merit of animals? So just taking you back a little bit quickly, this is just quickly giving you an impression of a cell. Of course, we all know that all living organisms are made up of cells, and just there's a lot of complex activity happening within cells, and that's important for us all to continue to remember, is that all of this activity are driven by the genetic material that's inside the nucleus. And it certainly is a complex system, and therefore it's not simple. It's not easy to just work out what's happening and how things work. But of course, the bit that I will focus on is the nucleus, which contains the DNA. So we're going to quickly have a bit of a look at the DNA. Some of you may have seen pictures like this before as well. So you have your cell, You have all of the genetic material that's in the nucleus, that's what we sometimes call the genome. That's all the genetic material. There's chromosomes in there, so for cattle there's 30 chromosomes. If you then unwind that chromosome, you have the DNA. And when you get to the next level down, you get these base pairs, what we call base pairs, and that makes up the genetic information or the coding that's there. So if you then take a piece of this DNA strand, that's normally a gene, so that can be longer and shorter, but that is the bits, and those codes there, and how it gets interpreted in division, and how it gets transmitted, and how a whole range of that bodies that I've shown you previously are using that, is then, of course, helping us to make proteins. And these proteins, of course, are in complexes, and that is what Brad basically looking at as well. But that can be enzymes, that can be hormones, that can be tissue. So all of it, right? So all of these proteins are affected by it, and therefore, if you have a defect anywhere in here, it could affect a protein here, but that protein could, and let's use the example of our genetic conditions, that protein may be critical for a specific development of a specific organ. And if that doesn't happen, an animal is not viable. And that is really the impact that we see is like NH is really a single base pair change in an animal. So a small little thing like that can have a big impact. So that's I just wanted to quickly get to that level so you just understand at what level we are working with in this case. So genetic markers, just stepping back a little bit. So what we try to do is we try to identify differences in the DNA. So that's what my previous slide was about, is really to say, okay, this is the DNA, so how do we work out the differences? And we do that through genetic markers. So genetic markers can be a gene or it can be a DNA sequence. So looking at the sequence of those base pairs, that's a genetic marker. It's normally have a known location on a specific chromosome, and it's used to identify individuals or species. So you can use that to get to that level of identifying individuals or species. Describe DNA variation normally that we can see. So hopefully if a marker is a good marker, it will show us the genetic variation, which are then displayed as a phenotypic or a visual difference in the animals. And normally this variation occurs due to mutations. So you see that there's a change in the DNA, and then of course, especially again, if we look at NH or nuclear, let's not worry about that. But in those cases, what happened then is that all of a sudden we have a problem and we try to find out where is the problem. Okay, so just again on to find or to create, to generate or create DNA markers, this is a slide really just quickly to show you that there's a whole range of different ways that scientists use different setups or different ways of identifying them and finding and creating these DNA markers. So there's not one single recipe that works, so depending on exactly what's the problem or if they want to identify, there's a whole range of techniques that they can use. So then going back into DNA markers themselves, as I said previously, there's ways of identifying, but there's also technologies that can be used. So there's dozens of different technologies that can create genetic markers. The ones that we really know well, and Angus members would certainly know them well, is we're using microsatellite polymorphisms. That is where our previous pair verification technology was based on that type of or those type of markers. And then now also single nucleotide polymorphisms are where we are now, again, our pair verification technology is now based on single nucleotide polymorphisms. So that's just two of the dozens that we use. Okay, so what we use genetic markers, and that's the main users of them currently in our system, is obviously for parent verification. That's a big part of what we do. Also for product traceability, so you can develop specific markers that you just use to trace products. Also managing genetic conditions, so we have several markers that are specific to genetic conditions, and then also now, more recently, certainly starting to get into the level of genomic selection. So we really start to use this to make selection, inform our selection decisions. So Angus genetic tests. So this is really just a quick timeline of, and I haven't added the specific years that they were introduced, but that is where we've been in the past just over a decade. Genetic Solutions had G-Star tests that they developed, and of course we had the 12 stars at the stage. Then after that they changed name and became Pfizer with 56 markers, EVPs, and that is where the technology being changed to become based on SNPs. Since then, in 2010, 2011, the 50K, HD50K for Angus product was released, and also, of course, then implemented where we were able to start to use that data in BREEDPLAN. Up to that point, that data was available, but it wasn't included in BREEDPLAN. Therefore, EPVs couldn't be impacted or influenced by the genomic information. So in that case there, that's where that started to occur. Also, the BCRC have developed SNP panels, so that information is there. We'll get back to that a little bit later. Also, Carel, Identity have SNP predictions. Of course, now Identity is GeneSeq, a company that people might know better by the name of GeneSeq, but they also have SNP or predictions that we can use and that we are in the process of getting that implemented as well. And then, of course, now Zoetis have developed or had a recalibration of the HD50K for Angus product, and I'm going to look a little bit at that in my talk. It really just gives people a feel for, because that's a question that I get a lot of times, is if I do a test, what impact will it have on my EPVs of my animals? So we'll have a look at that shortly. So just going back to SNPs, so that's the technology that we use now for our genomic test. So that's really where things have moved to, and already we're starting to hear that really our 50K test might not be the technology for the future. We may go to whole genome sequencing, etc. So that will be interesting. Of course, depends on the cost. So there's quite a few implications, but that will probably be the area where we will go to. So, of course, for single nucleotide polymorphisms, we just refer to them as SNPs, of course, pronounced as SNPs. That's more for humans, but I suspect that will be virtually the same for cattle as well. We have probably a SNP for about every 300 nucleotides. So for every 300 base pairs, on average, you will have zones where there's more, but on average about every 300 nucleotides. So that means that there's about 10 million SNPs in the genome. But those are also found mostly between genes, so it's not in the gene, they are between the genes. So they can give us an indication. If we know which SNPs we're looking for, we can actually, if we know what the function of the gene is and where that gene is, we should be able to find SNPs that we can then associate with some of those genes. And that's probably easier said than done, because there's sufficient work done to try and find them, but it's not that simple. And of course, in a lot of cases, you don't have one gene that influences a lot of your production traits, especially. Of course, for colour and those, it's a lot easier, but the production traits, a lot more genes influence it from different aspects. Okay, so that's just a quick rundown on SNPs. Just to explain that, so what you have your chromosome, you have your DNA, and then what happens is that if this is your normal one, what's happening here is all of a sudden this base pair here got replaced replaced by that base pair there. And that's really the size of the change that happened with NH. So it's a single base pair change that caused NH and those animals to be dead on birth. So it just showed how big an impact that something like that small could have. But that's a very, very simple explanation of what a SNP is. Okay. So the Illumina 50K chip is what I mostly use at the moment. There's, of course, a lot of the companies, and I don't know if Zoetis have a totally different, Illumina to develop a different chip for them, but I know that GeneSeq has extra chips that they've put on, or sorry, extra SNPs that they've put on their chip. So, but in this case, what's happening is that, as I said previously, there's about 10 million SNPs available for us. And what we have here is we have really 54,000 to work with. Where I said previously, there's about every 300 nucleotides there's a SNP available. With that number of SNPs over the whole genome, that is roughly 67,000 base pairs apart. So you can see how the spots, the number of spots where we really should have the ability to fine map it, we're not even close. Of course, if you want to have that many SNPs, the 10 million SNPs on the chip, that I think will be a little bit cost not going to make it very efficient for us to do. So we have to just at this stage work with those. You do get we have 50,000 SNPs or 50K chip as well, which is a bit more expensive again. So that's just how that works. This is basically the chip. This is some of the software that they use, and without going into too much detail, so every one of these dots represent a specific location on the chip, where they then see how that specific feed actually react to chemical sets, and also what actually gets attached to it, and work out whether or not which SNP is actually sitting in that location. So that's just a little bit of information. So using genetic markers in BREEDPLAN, so the basic principle that we start off is that we say that markers does explain some percentage of the genetic variation. And that is where we start off with. So if that is the case, then it must and it will increase EBV accuracy, because what the EBV accuracy does, it gives us an indication of how much knowledge do we have about the information and that specific trait. And of course, if we can add additional information through markers, then that should increase the accuracy. If we then have more accurate EBVs, it should be able to reduce our generation interval. So, especially with markers, you can actually use them on very young animals. So, therefore, you can get to a point where you can start to select animals for maternal traits or even carcass traits when they're very, very young. You don't have to wait until a lot of their progeny, for instance, gets killed, and then you can really start to identify them and identify them earlier on. Also, with all of that, especially in generation interval, the accuracy of selection will make our genetic progress quicker. So that will contribute then to a faster genetic gain. Right, so then just quickly going into the genomic predictors of how we include them in BREEDPLAN. So there's two steps involved. So the one step is the discovery step, and then another step is the calibration step. So what's happening in the discovery step is that's normally in genomic companies. What they have to do is they, of course, have to have big numbers of SNPs. Again, try to cover the DNA as much as they can so that they can get a very good understanding of which genes are impacting on a specific trait. To do that, of course, they have to have big data sets, and of data sets I mean animals that has their traits recorded and also have genotypes. So it's not good enough to just have an animal that have a measurement. They an animal also have to have genotypes, just like Brad indicated as well. It's important then to have an understanding or knowledge of the genetic information or the SNPs that's there to help us understand which genes are affecting that trait. Then, of course, what needs to happen is you need to analyze that information to determine which SNPs influence which traits. So you then start to do an analysis to work that out. And once you've determined that, you produce genomic predictions. So that can be the Zoetis MVPs, or it can be the GeneSeq MVVs, or Genity MVVs, or the CRC DGVs. So they're all basically the same thing. And then, of course, there's the calibration step that AgBoo does. So what they then do is they take these genomic predictions and estimate their effect on the BREEDPLAN traits. So they say, okay, we have these BREEDPLAN animals that has been BREEDPLAN reported that has EVVs, and what is the effect of this on the actual performance of animals? So then they do that analysis. But to do that, they also need independent animals. So they need animals that have not been used in this discovery step. Because if you use those same animals, you'll come up with the same answer as what the genomic companies have done. That's a challenge. To find animals that are totally independent is one of our big challenges. That is where the site benchmarking program is certainly helping us and will continue to help us to try to help in this step here. So I'm going to focus a little bit more on the Zoetis HD50K discovery step. So what they've used is they've used the 50K snippet. They've also used a global data set. So Zoetis have a data sets, I suspect, that they can use for discovery. But also they've used Angus Australia EVPs. But what we've said is for us to allow the second step, or that step that AgBoo needs to be involved with, we were going to exclude it. We did exclude the site benchmarking progeny. So we decided to keep it out. There's other progeny animals that we kept out, progeny test animals that we kept out as well, out of this discovery data set that we supplied to Zoetis. And then what they did is Zoetis were able to develop MVPs for 22 traits. Of course, you will say that there's not 22 MVPs available, and that is where the global data set comes in, that they've used other data to be able to get some of those information on some of those traits as well. So then the calibration step, if we go in a bit more detail there. So EVVs we use for discovery, of course, as I mentioned before. The grand progeny we use for calibration. So to create that distance between the animals that were used for discovery and the animals that were used for calibration, we make sure that we don't use the animals themselves or their progeny, but only the grand progeny to create that unrelatedness. Of course, our challenge again here is few other unrelated animals are available, like totally unrelated. That is a challenge that we just have to deal with. So carcass traits that were used in the calibration were between 1,000 and 5,000 animals were available for calibration, which is a good number, but of course if we could get that more, that would be better. And that is again the site benchmarking will help us, the site benchmarking program will help us to increase that a bit more. And for the other traits, that is money or growth traits and calving ease and gestation length, etc. We have more than 40,000 animals for those calibrations there. So there's plenty of animals to use for that. Efficiency is still a big challenge because we only have limited numbers there, and of course it's a very expensive trait to report, and that's part of the reason why we say that it is a critically important one. We of course continue to say that it would be great if we could use genomics for our very expensive and difficult to measure traits, but at the moment our biggest problem is that we don't even have enough data there to actually get to a level where we can say that we can get that genomic values to be a very good indicator of the performance of animals at that level. So, and then what has happened with that calibration is AgMove came back and said, listen, we've been able to calibrate 14 MVPs for blending. So 14 of the MVPs of those 22 that I indicated previously is actually used by BREEDPLAN and contribute to the EBV equivalents. Peter's chasing me now. Come on. I'm quickly going to go through the genetic correlations. So this is really just giving you an indication. Genetic correlations, what that says is that's the genetic correlation between the MVP and the EBV. So that's giving us an idea of how well, and this is good. This is really, there's quite a few of them that are in a very helpful level. We just a little bit still disappointed that we couldn't get better levels for muscular fat. But these are starting to become really handy correlations. And of course you will see that most of it has, this was the 2010, 2011 product. And this is now the newest release. So we are very excited and looking forward to how this will of course continue to develop. So, and that's really what I'm going to look at in my next slide. Is I'm quickly going to give you a visual impression of what the impact of testing is. And I don't have time to go through all of the tracks, so I've selected 2 tracks that I'll look at. So the one is looking at mature cow weight. So you can see that it has a high correlation, genetic correlation, and I've also used intramuscular fat because it's got one of the lowest genetic correlation. Just to give you an impression of how that influenced the EBVs that we did. So in this case, there were some animals that were blended, and some animals don't get MVPs or animals don't get MVPs for every trait because it doesn't agree on which SNPs are available. But in this case, that animal wasn't blended. So the bottom line is that we're looking at the mature cow weight accuracy. And so this is the unblended accuracy. So that is before what you would normally have in BREEDPLAN if you didn't get the animals tested. I should say this is for about 8,000 animals that we currently have tested and where we have MVPs available. So that is the animals' unblended EBVs, and then that is the blended EBV accuracy. So of course the accuracy. So what you can see is that if an animal, for instance, had a 0 accuracy or very close to 0 accuracy, just by testing them for the 50K, that animal's accuracy would go up to about 58, same as, 59. What happens then is as you come up, let's say an animal had a pre-blend accuracy of 20%, that would increase to almost 60%. But as you go up here, you can see that once you get to this level here, the testing don't really contribute extra information, so the accuracy don't really increase. Having said that, you'll see in a few slides that the MVPs still have an impact on the EBV, and that's an interesting that we will look at in a minute. So, but that's really just giving you an impression that as the accuracy, and of course this unblended accuracy here depends on the pedigree information, it depends on the performance that has been done, etc. And the more performance you've done, the higher, and performance on related animals you've done, of course, the higher your starting accuracy would be as well. So if you're a herd that does a lot of performance recording, you'll probably sit in this area anyway, and so therefore your benefit that you'll get is relatively small. The other bit that's very important, small herds would certainly benefit because they could potentially sit in this area here, and therefore they will get more benefit from testing. Right. So then just looking at the physical EBV. So again, unblended EBV on this side and the blended EBV. So let's say an animal that had a mature cow weight EBV EBV of 100, sitting over here, it could go up to as high as 140, or it could come down to about 60 odd. That's a dramatic impact. But also remember that that is linked to the accuracy of the animal when it starts. So as there's more information, it does the possible impact decrease. I'll show you that in the next slide. The other interesting bit is just take a note, because that is if you had an absolute equal opportunity to go up and down, of course all of these values would sit around that line, like that. What you would see here is that at the lower level, more animals are sitting below the line, and at the higher levels, more animals are sitting at the top. So that means that there's certainly a correlation there, and therefore it is actually picking the animals with the lower EBVs and putting them actually further down. Right, so that's just an interesting observation as well. So this is the other interesting one where, in this case, what I've done is I've looked at the unblended accuracy and then also at the EBV change. So when an animal got blended versus unblended, that is this change here. So the interesting bit, and that is the real point I want to make, is animals sitting at, unfortunately this is good, so that must be 80, 60, 40, 20. Animals are sitting at about 60% accuracy before you test them. Could of course go down by as much as 40 or up by as much as 42 or 43. As the accuracy of those animals increase, the possible change decreases. So what the software says is, listen, I already know a lot about this animal's EBV, so I'm not going to allow the MVP to have such a big impact. So it's actually reducing the possible impact of the MVP on the EBV, and that's what you see here. You start to see that at 100 you don't get animals that change 20 or 30 kilograms up and down. And that's exactly what we would have expected. So it's very good to see that that's occurring. Then also this was just really a quick one, also to just say, does everything make sense? To me it does. Because of course the MVP in this case, what we have is we have a very high MVP here and a very low MVP there. What we've seen is at the low level, the EBVs actually got pulled down, and at the high level they actually got pushed up. And these animals here, you could say, well, why aren't there more animals filling in those blocks? And I think that's to do with the accuracy. So again, it limits the possible change, and therefore what it does, it limits animals to pull them into that very low up or down change in there. Right, so that is the cow weight. I will take the opportunity to do just a few more, but it's almost there. So again, as I indicated before, the genetic correlation here is dramatically lower. So therefore you will see that the benefit that you get accuracy-wise is reduced. And that's also what we expected. So you'll see that coming into 40 and 50% is very, very little benefit, accuracy-wise.
How do we know, because that correlation is so low, whether the problem with scan is more a problem with the genomics?
At the moment, that is a big challenge. And of course, and that's one of the best that we really depend on the side benchmarking moving forward, that we really want to get that information starting to come in as well. There's no easy answers to any of those. Because the EBVs currently, a lot of them are based on scanning, and scanning has its own challenges as well, just as we heard from Peter and from Alex this morning, that there's challenges there. And so, but this is one of the technologies that we certainly want to find out more to try to address some of those challenges. But that's, I'm not going to repeat everything, but you understand now the concepts. We have the same situation here where at the lower levels, animals mainly move down. The higher levels that move up. It is interesting to see that you can get up to 1% up and down relatively even. Okay? So then again, the accuracy, so again, let's just show exactly the same again. So when an animal sits at about this 40 to 50, you get a lot of spread still. As you go closer to 100, that spread decreases, and in here you can see that you can get as much as 1% up or 8% down, extra spread just around the in-betweens. So that's good. Then also, same deal again, that's what we expected there. Right. So quickly just multi-source blending, that is the next step that's going to occur. Angus supplied us software to blend genomic regions from several companies. So we can now also, we can actually now also blend the BCRC EGDs and also the GC MDDs. But that is going to be implemented in the Genomic Replanting analysis. So there's quite a bit, I said quite a bit, there's probably about 3,000 animals with DGVs and MDVs that will be included in. They're mainly older animals now. We had a project in 2010 and another one in 2012 where we got industry animals tested under a whole range of different arrangements. And those animals that will now be able and be rolled into replanting as well. So the future, currently we have a two-step process, as I explained previously, and also the blended information only impact on the animal itself. So only the tested animal gets the benefit of the test. What we certainly are waiting for, and Ag was frantically working on developing software that will basically use both of these steps and include both of these steps in replanting analysis. So you'll have a continuous check on which SNPs are potentially contributing to an animal's higher production, and therefore that calibration and discovery occurs every replant month. And of course, the benefit of that then also will be that it will impact related animals. So that's just a quick rundown on some of the things. We certainly are looking forward to a lot more development, especially we are really pushing and asking and begging Agro to continue to really work hard on getting that work done as soon as possible so that we can start to test it and see really what impact it has. So thank you very much. I hope that give us your answers, and you will probably be saying that's it. Okay, thank you very much.